Mary Lyon proposed that one X chromosome in each female cell becomes inactivated. This process, X inactivation, is random and happens separately in individual cells. Years earlier, Barr discovered Barr bodies. A Barr body is an inactivated X chromosome that appears as a condensed, darkly staining structure in most cells of female placental mammals. Barr bodies are what Mary Lyon was referring to when she proposed that one X chromosome becomes inactivated. The Lyon hypothesis also proposed that if a cell contains more than two X chromosomes, all but one will be inactive. This hypothesis led to discovering more about the expression of X-linked traits and X-linked genetic disorders. Here are a few examples of Barr bodies in humans and the resulting genetic conditions from these inactive X chromosomes. As a result of X inactivation, female placental mammals are functionally hemizygous at a cellular level for X-linked genes. The term hemizygosity is the possession of a single allele at a locus. Males are hemizygous for X-linked loci because their cells possess only one X chromosome. In females that are heterozygous at an X-linked locus, around 50% of the cells express one allele and 50% express the other allele. Also, proteins encoded by both alleles are produced but not within the same cell. A great example of X inactivation are tortoiseshell cats. A single X-linked locus determines the presence of orange color in the cat's fur. The process of X inactivation takes place in early development. After the X chromosome has become inactivated, it remains inactive in the cell and in any following somatic cells. Neighboring cells tend to have the same X chromosome inactivated, which produces a patchy pattern. The mechanism of random inactivation is two steps. First, the cell counts how many X chromosomes are present. Second, one X chromosome is selected to become the active X chromosome, and all others are inactivated. Also, an important gene in this process is known as the X gene. This gene exists on the X chromosomes that will be inactivated. Thinking back on the example of tortoiseshell cats, there are two possible alleles at this X-linked locus. The male cats are hemizygous and may either have the allele expressing black or orange hair. Females, though, they may either be black, orange, or a tortoise pattern. In the orange patch, the X chromosome with the orange allele is active, whereas the X chromosome with the black allele is inactive or represented as a Barr body in the picture. Vice versa, for the black patch, the X chromosome with the black allele is active, and the X chromosome with the orange allele is a Barr body. So what about this question? The answer goes back to the principle that X inactivation is random. The cells at random would inactivate either the X chromosome expressing the black allele or the X chromosome expressing the orange allele. Cells derived from the original cell would continue these patterns in inactivating one of the alleles. They would then group together based on the expressed allele, creating different colored orange and black patches. Here's a visual of possibly what the clone cat would look like. As you can see, the cells with the same inactive alleles group together to form patches of black and orange.